So it's really great to be here. I've been in this meeting a couple of times, and I think it really has the best to offer of genome sciences, which is combining the newest technology with interesting questions in biology. In my lab, we don't do the types of things that Jeremy talked about. We rely on those things and use them on a day-to-day basis that really accelerate everything that we do quite dramatically. And I'm actually thrilled to hear Jeremy be excited about these genomes. But I'm going to say some positive things and negative things, is that these are some of the species that are proposed or being developed as energy crops, right? So for the obvious reasons is that they are positively gigantic. So these are grasses. This up in this corner is miscanthus. Down here we have sorghum, and here we have switchgrass, right? And these are really, to me, very interesting organisms. And by virtue of their attributes, they become potential energy crops. But they can be, rather awful to work with if you think on an experimental level, say in the laboratory, right? So you would need the space to actually cultivate all of these plants. They tend to have long life cycles. They can be difficult to cross. In this case, in miscanthus giganteus is a sterile triploid, right? So if you want to do genetics, you have to go to the progenitor species. Sorghum is actually fairly tractable as far as grass genomes go. This is an annual, the other two are perennials, and it has a diploid genome, and Jeremy mentioned the sequences available. And switchgrass here is an obligate outcrossing tetraploid or octoploid. All right? So that brings me to pointing out this very teeny tiny plant in my son's hand here, which is a Brachypodium distachyon. Right? So for the reasons that discovery can be very difficult, at least on an organismal level, we're working with this species. And so we can see it simply compared here next to Arabidopsis, which would be the real gold standard for a model system in plants, is that it similarly has a small stature, it has a very short life cycle, it's easily transformable, and it has a really great genome, and it's super exciting that another version is coming out. It already started out quite nice. And it has a close phylogenetic relationship to other grasses. So here's brachy up here, and is similar to barley and wheat, rice, switchgrass, sugarcane, sorghum, and maize, and there's about 150 million years of divergence from dicot species like Arabidopsis and soybean, for example. So there are lots of attributes that exist within the grasses that aren't within dicot, so this serves as a nice functional model. Okay, so when we think about growth, in my laboratory, we're really thinking about cell wall biosynthesis. Okay, so what are the important traits? One is that we just simply want more biomass, right? And this is where it's happening in the cell wall. So, right, so here are my people. We always learn that plants, the distinguishing feature of plants is that they have a cell wall, but it's just that puny little animal kingdom, right, that doesn't have a cell wall, maybe protozoans. And so they make this primary wall, which can be elastic and part of a growing cell that can change shape and size. And then some cell types, when they stop growing, will fill in with this secondary cell wall, which is shown here in brown. All right? And so here is a stem cross-section of a brachy plant. And what I want to draw your attention to, are a couple of things. So these circles here, so it's all circles, but these distinguished circles here are vascular bundles, right? So this is the vascular system. Similar bundles appear in all the grasses. And these larger cells at the bottom are the xylem. So this is water up. And these smaller cells here in this section are the phloem. So this is water moving down. And then other notable cells in this space would be these very thick walls. So these are fibers. So these are primarily structural cells. And so what I'll also point out here is that over 3/4 of the total volume of these cells is simply secondary cell wall, right? So when I'm thinking about biomass that you collect from a plant, most of it, or a grass, most of it's all going to be stem, and most of it's going to be secondary cell walls, all right? So this is the biomass that offers opportunity. And so the primary wall is cellulose, hemicellulose, and pectin, so almost all fermentable polysaccharides. And the secondary wall, we add in this fraction of lignin, right? So you know about lignin is that this is not a necessarily usable fraction unless we're going to burn, perhaps. Is that this is providing hydrophobicity and structure, but it's a chief recalcitrant to at least biological conversion of plant biomass. Okay? And And again, the polysaccharides are simply made of sugars that we can then convert into, or use microbes to convert into fuels. Okay? So the important things are lots of biomass and having biomass meet a particular quality standard, right? And this wouldn't be any different from any other crop species, okay? And which leads me to a scan of an actual slide from my master's thesis research a while back, where I was studying sources of variation in cookie quality, right? So I actually used to be a plant breeder. And every crop has an assay in the end, which is this going to meet quality standards? Because you're plugging in something in high volumes in industrial standards. And what bakers want is at least they're using soft wheat to bake cookies, is they want a big cookie. So there's my master's project. I baked cookies, and I had a caliper, and then I measured how wide it was, right? And I knew what the genotype was and where it was grown. And so I ate a lot of cookies. And a little known fact is they called me the cookie man when I got my master's degree. So this cookie here is better than that one, right? And so they want a soft, widespread. If it was hard wheat grown in the western U.S., you would bake a loaf of bread and measure the volume. Or if it's potatoes, you would chip the germplasm or the varieties and see if it came out to be the right color, right? Because these are very complex traits, and you're plugging them into industrial scenarios. So we have an assay that we use for feedstock quality, right? And there are many similar assays, at least on the enzymatic level, is that we grind up the plant biomass and feed it to an ethnologen, right? So in this case, it's Clostridium phytofermentans, right? So here we just have some sorghum, which is stained, the cellulose is stained, and a DNA stain for showing the microbe. And it's, I'm not a microbiologist, but this is a bit of an omnivore, so any polysaccharide I've mentioned, this can metabolize it. So it secretes hydrolytic enzymes and can consume both 5 and 6 carbon sugars. So the idea is that it might prefer some substrates over another, and that turned out to be the case. So we took sorghum, so field-grown sorghum varieties, and we have wild type and mutant. And you'll notice this change in color, which these are referred to as brown midrib mutants, and these were identified a century ago in maize. And these are incorporated into modern varieties of sorghum and maize, and that's used for when you would, say, chop it up and feed it to livestock. And because these are more digestible. And they're more digestible because they have mutations in the lignin pathway. All right, so if we simply measure ethanol when we feed wild type or 2 different mutations in that pathway, or the double mutant, we get a big bump, or in one case, twice as much ethanol when we feed the double mutant from sorghum. Right? So we have what we think is at least a workable assay that uses an actual ethnologen. And we're applying this with material from other programs in miscanthus and sorghum and rice and barley in other people's mapping populations. And we're also using it in our own mapping population. This is actually one developed by Dave Garvin, USDA in St. Paul. And what it simply is that we have 2 parents, right? They're crossed, and then through successive generations of inbreeding, we come up with a population of inbred plants that are 50% mixtures of each parent, but different mixtures. So we're looking for co-segregation or associations with genotypes and phenotypes. And in a very small amount of space, in a very short amount of time, we can go through one whole cycle of this population. So 3 or 4 generations, or 3 or 4 separate trials, just on one small bench in the greenhouse in 1 year. So, which brings us to our QTL map, right? And so what we have are just the 5 chromosomes of brachy, and this is a linkage map without the markers noted. And we measure the very obvious traits, so the height and weight. So these are important biomass traits, and also digestibility. And so what you can see is that so there's a blue and the green bar here, which means there's a QTL. There's an association between that region of the genome with stem weight and stem height. Okay? And we also have one on the top of chromosome 2 for digestibility. So, and then if we were to divide up the members of the population into all of those that have one allele at that top of chromosome 2 versus all of those that have a different allele, there's an 8% difference in ethanol yield on average, right? Which is a huge bump, right? So plant breeders are trying to go for 1 or 2% improvement every year. Is that we have an allele which is an 8% change, okay? And so it's at this point that we are absolutely even relying very heavily on all of the sequencing done at JGI. So I saw Wellington give a talk a month ago at a different DOE meeting, and this is the so-called QTL junkyard, I think is what he called it. So if we don't have sequence, we don't even know what genes are in that space, right? Which is definitely not the case now, is that, so out of the more than 25,000 protein coding genes, there are 139 in that interval, right? So we've reduced the number of candidates, and then so they would necessarily need to be polymorphic between the two parents for it to have an effect on the trait, right? And this just initially came online. It's developing by the day. Sean Gordon is working on this, who's speaking later in the session, is that we can look in the browser and the track and say, yeah, so here's an annotated gene, and there are two polymorphisms here between these two parents, and this results in a change in amino acids. So this would be a candidate at this point, right? And then we also rely heavily on expression profiling, right? So that atlas is really useful. We've done this with Affymetrix arrays that were supported from the feedstock genomics program. And so stems have lots of secondary walls, and the transcripts are highly abundant in that tissue as opposed to, for example, leaves, which don't have very much wall or secondary wall. So we are expecting to see profiles like this. So this is expression abundance. This is a known gene, so it's very low expressed in leaf, root, and very high in stem. So if we have candidates in that interval that look the same, then they're even better candidates or things to follow up on. And then, of course, annotation. So there are a couple of genes out of those 5 that, one's a facilin-like arabinogalactan protein and one's an arabinofurantosyl transferase. So these are things that have been shown to be associated with walls. They're polymorphic, and they co-segregate with our trait. So we can follow those up with more forward genetics. And we can also, so we have differences in plants, and then we're going to just keep looking for recombination events and prove that the polymorphism causes the effect. Or we can put it in a reverse genetics pipeline, right? So where we take genes that we have a hypothesis about their function, and then we can test to see what happens when we perturb those in the plant. So one resource are tDNA collections. So there are several labs that are developing these in brachy. One is John Vogel's lab at USDA, so there are about, I'd say, maybe up to 20,000 different insertions. These are cataloged and sequenced, right? So you can, if there's an insertion in your gene, you can order that. We commonly use artificial microRNAs that are designed to target transcripts. If we're working with transcription factors, which we often do, we overexpress those, tagged to repressor domains, or there's just plain old overexpression, right? And the transformation efficiency is extremely high, at least relative within plants and brachy. I would say second to tobacco and Arabidopsis, but it's improving very often. So what have we been able to do? I'll just show a couple of several is one are to identify lignin genes, right? And these are so that we can study the regulation downstream, but we've made our own brown midrib brachy mutants, right? So the ortholog to this gene in sorghum, if we silence it, we get a brown midrib mutant in brachy. And interestingly, they tend to flower late, which is something that's not understood about why that would happen. But they're about twice as much biomass. One is that they're just flowering later. And then they also turn out to be more digestible. So similar observations in sorghum, right? So we have brachy mutants that have more biomass and they're more digestible with a mutation in a known gene. We've also pulled out cellulose genes. So this is polarized light microscopy. So again, another cross-section of a brachystem. And when you see the white light or when you see the walls, that means that's an observation of crystalline cellulose. And in our mutant, we have a very, very little crystalline cellulose in that stem, right? So it suggests this is a gene involved with making that secondary cell wall cellulose. Okay? So where we want to bring this is understanding the regulation of those genes, right? So the top model are transcription factors, so each one of these rectangles that are NACs, which are plant-specific family, and MIBs, which are common across life, and these are regulating hemicellulose, lignin, and cellulose genes. So what we want to do is, what does this look like in grasses, right? So they're distinct in their anatomy for many cell types and at least the way they're arranged in the stem, and also on some levels the composition, right? I showed you some of the lignin and cellulose mutants, so we've populated this space with targets. Now we want to understand the transcription regulation. So we've begun to build a full-length transcription factor library, right? And Jose Pruneda-Paz, who's going to talk in the next session, will describe a very nice or nearly complete collection of Arabidopsis, and it's powerful, a really powerful tool. So we've been cloning. We're also doing this with Todd Mockler at the Danforth Center. Steve K. in Southern California. Recons contributed a great many clones, and then the JGI is generously offered to sequence part of the population, okay? And so this is a community resource, and you can use it for various things. And I won't get into, and we're using them in yeast, right? So we're doing one hybrid screens where we are looking for an interaction between our brachy promoter fused to a reporter, placed in yeast, and then we add in, individually the transcription factors. And if there's an interaction, we have a simple visual assay. Again, Jose will show some more advanced approaches using these screens. And so what I've added to the model this time are other genes from other grasses, so OS rice, ZM maize, and these aren't connected to those downstream genes because they've been tested in Arabidopsis, so in a heterologous system. But this switchgrass and maize genes have been identified to repress lignin. And what you'll note is that they're similar colors, so those are the closest orthologs, and that perhaps have similar function. And we found something similar as well, is that if we see the matching colors here, is that we found these HB proteins that are same as been identified in Arabidopsis, and the same thing with all these MIBs and these NACs. And where there is an overlap, I think there actually is overlap in Arabidopsis. It just hasn't been discovered. So the structural enzymes and the regulatory proteins, I think, are largely similar, actually, across dicots and monocots. But there are key distinctions within grasses that make them, you would have to work with them and not with Arabidopsis. And this is an example of one where we took one of those NACs and we make the repressor, which is effectively similar to the wild type. But if we overexpress it, we get this little brachy bush, right? So it doesn't, we can coax it into making a flower, but mostly it just continues in this vegetative state. Which brings us to a key difference between Arabidopsis and grasses, is that Arabidopsis doesn't make a stem until it's flowered or transitioned into flowering. But grasses are making stems and then can transition into flowering after that. And then, interestingly, we have this developmental phenotype here where there are twice as many vascular bundles in the mutant as there is in wild type, right? How that translates into digestibility and the amount of secondary wall, we don't actually know, right? But there's a different relationship between vegetative and reproductive stages and cell walls and grasses than there appears to be in Arabidopsis. Okay? And another gene, which is a MIB transcription factor, if we overexpress it, and it's not necessarily apparent in this image, is significantly larger and it has more walls. And the repressor line, the dominant repressor, is also dwarf, but it's a very different class of dwarf. Is that it has the same number of leaves, it has the same number of internodes, it flowers on the same day as the other plants. It's just a dwarf, so the cells actually don't elongate. And that's likely due to a structural effect. So again, a stem cross-section, and this red stain is for lignin, and the bundles are largely similar. These might be a little more faint, so that would mean less lignin in the bundles, but the fibers have considerably less lignin. Their thickness implies there might be secondary wall, but they don't actually lignify. And this is perhaps why the plant doesn't elongate, right? We haven't tested this, but these are certainly going to be more digestible, but that's not going to get you very far when your plant is 10% the size of a wild-type plant. But I think this becomes an effective tool, right? So now we have these genes which we use really blunt force approaches to find their functions, and then we can go and look for more subtle variation and natural accessions, especially all these that have been resequenced. Okay? So the last project I'll talk to you about is more about the other kind of growth. I'm saying growth to us is just making more biomass, is that this is more of that dynamic change where I can see my plant is actually growing. Some of the maize plants are getting taller in the field. And this goes to a point, so this is in the evening here, and this model is reasonably well worked out in Arabidopsis, where most of the growth, or at least hypocolloid elongation, occurs within a very narrow window of time. Okay, so the hypocolloids we elongate in the end of the evening, continue to do in the morning, and then they're repressed by 2 BHLH transcription factors, and then those degrade at night, and then the clock maintains that repression. And this has been observed in a lot of dicots in soybean and I think poplar, for example, where there is a narrow window of time where most of the growth occurs in plants, right? So this, to me, is a really fascinating question and also perhaps an opportunity. So think of it as on the planet, there's this 5 or 6 hour window where most of the biomass, plant biomass accumulates, right? That's something that I want to understand. So we developed an imaging system to look at growth in brachy. And so what you see here are 5 leaves that emerge from this pot, but this pot is actually in the dark. So we have an IR bulb and we have an IR camera that's watching these plants. So we're taking time-lapse photography, so we can't see IR. Our eyes don't have those photoreceptors. Plants don't have photoreceptors to see that wavelength either. So they have no perception of that light. And then we can perturb the conditions to figure out when that rhythmic growth happens. And you can see, at least under these conditions, if we crop out that leaf, we can see that there appears to be a rhythm where there's fast growth here and slow growth here. And so I've talked to you a lot about stems, and stems are hard to do photography of in grasses because there are leaf sheaths wrapped around the stem, so you can't observe a naked stem moving up until that last internode. But what we have been able to do is track the nodes, and it appears as though that stem growth is the same as leaf growth in grasses. This is definitely not the case in at least Arabidopsis. So we've gone in and tested different conditions. So we always first look under light, dark, hot, cold. So that would be this: the lights are on, the lights are off, it's warm or it's cold. And then in the first of 2 movies, which I'll show you, are plants which are released under constant conditions. So what would happen in Arabidopsis is they would maintain a rhythm, right? So the circadian clock persists. We have rhythmic gene expression. We also have rhythmic, elongation of the hypocotyl. Okay, so if you YouTube, my plants grow fast when it's hot, or you might find this video, but I suspect there actually aren't very many videos of plants on YouTube. So and the movie's annotated in the top. So again, we have, excuse me. You can see that during the day, they're growing very quickly, and then when it's cold and dark, they slow down. And then we're going to transition into lights are always on and it's always warm. What you can see is there are actually no changes in growth rate, right? They're just growing fast. And so this was really startling when we found this. And what it means is that, so I was trained as a circadian biologist, and now I work on the one trait, I think, in plants that has nothing to do with the circadian clock. Because there is no clock-regulated growth. We've gone and looked at time courses, and we have clock-regulated gene expression, but the growth rate is not affected by the clock. Okay, we've also tested, so in the constant dark or the constant light, there's no rhythms. There are no rhythms. If we toggle light and dark, but we keep a constant temperature, there's a small effect, right, by light-dark. But basically, what we find is an effect of temperature. So if we grow them under the constant light or constant dark, for that matter, and then we toggle warmth and cold, you see basically it looks like a regular day. I'm not very good at that. Okay, so same thing, they're growing fast during the day and slow during the night. Now the lights are always on, and they're tracking changes in temperature. Okay, so everything that we are observing in brachy, and this turns out to also be true by another group that's looked at this in rice and maize, is that it's temperature. So temperature is driving growth rate in plants. So, and so good grief, now all of a sudden we're working on the temperature sensor. But that's not actually true. What we want to do is integrate this information with the cell wall biosynthesis. So what we have no idea what's happening, and this is true in Arabidopsis for that matter, is that we have these changes in structures, we have growth, but is there cellulose? Is it increasing in abundance in one of those windows or not? Or are we just talking about cell elongation? It's not exactly clear. So we want to integrate these growth signals, which are generated by changes in temperature, right? And we can talk about how that would actually happen, and with actual cell wall biosynthesis. Okay. We've had a good collaboration with microbiologists at UMass, people that work with Clostridium phytofermentans, Sue Lachine and Tom Warnick and Jeff Blanchard. Dave Garvin and John Vogel contributed lots of resources and reagents. And the growth work is done with Steve Kay and Ben Cole, and some transcription factor work with Jose Perneda Paz. and I also already mentioned Todd Mockler, and generous support from the DOE for all of these projects. And then most of the data is provided by Pubadu, Handa Kumbura, Dominic Matos, and Scott Lee. So thank you.
Thanks, Sam. So climate change is going to help us a little bit, right? We'll get hotter, the plants will grow faster, we'll be able to burn them better. Great. Any questions?
Hey, nice talk, Sam. So I'm wondering if your conclusions on the growth would be different if you use mature plants. So does ontogeny play a role in perception, whether it's cold or light or?
So on plants that are older?
Right.
So we don't know. We don't know that yet.
First, do you think that the temperature, do you think there's actually a temperature sensor, or do you think it's just enzyme kinetics? It's just higher kinetics at higher temperatures?
Yeah. So perhaps heat is simply warming everything up, and that's a catalyst, and that's what's synchronizing everything. So there's one nice experiment where the different enzymes were tested, growth-associated enzymes were tested in vitro, and they behave very differently in response to temperature, which suggests there must be something that's integrating those signals. There must be a temperature sensor, right? But it's not like a phytochrome, which is simply changing conformation due to the presence of a photon, and then it migrates into the nucleus. We don't know what's sensing temperature in plants necessarily. So I do think that something is very likely integrating. And we see a linear increase. So if we test in between temperatures, it's a very linear response. But I wish I knew.
Yeah. Second question is, do you see an increase or a correlation with digestibility and pathogen susceptibility? So less lignin, higher susceptibility, basically?
The literature actually now is suggesting, this is what everybody said in the beginning, is that we're going to knock down lignin, and the plants will fall over, and they'll be susceptible to disease. And that doesn't really happen. And there are actually lignin mutants that are more resistant to disease because of spillover into the pathway activating salicylic acid. So I think it's very likely to go either way, and it also turns out to be highly background dependent. And that's probably the effect of that induction of immunity. Right? So maybe the lignin mutants are actually dwarfed because they're totally freaked out because they think they're increase their defense mechanism.